What if the true key to unlocking Mars lies not in shielding, but in symbiosis? For decades, we've poured our efforts into constructing walls against the cosmic storm, barriers of metal and plastic, destined to falter. But imagine a living shield, a microscopic army forged in the crucible of space itself. Can we truly entrust our lives, our very DNA, to engineered organisms, banking on bacteria to brave the radiation that would otherwise transform our astronauts into ghosts? The red planet beckons, an irresistible siren song, but the price of admission may be a leap of faith unlike any humanity has ever contemplated. But imagine, what if life itself served as our shield? Could the very organisms we associate with fragility become our armor against the harsh realities of the cosmos? Before we venture further, what's your wildest guess? Subscribe to uncover the full truth. Radiation exposure in space, a silent and invisible killer, can elevate an astronaut's cancer risk significantly. But what if we could somehow harness the extraordinary resilience of extremophiles? Consider Deinococcus radiodurans, affectionately nicknamed Conan the Bacterium, a microbe renowned for its ability to withstand radiation levels lethal to humans. What breathes such resilience into life? What grants certain organisms the ability not only to endure, but to flourish in environments that would instantly obliterate others? Behold the extremophiles, nature's own daredevils, life forms that scoff at conditions lethal to most. And among these hardy survivors, Conan the Bacterium can withstand radiation doses up to a staggering 10,000 Gray. To grasp the magnitude, a mere 10 Gray is fatal to a human. Conan's secret lies in a multi-layered defense. Multiple copies of its genome serve as backups, ready to deploy should disaster strike, while highly efficient DNA repair mechanisms swiftly reassemble shattered DNA fragments. But Conan is not alone. From nuclear waste sites to scalding hydrothermal vents, other bacteria and Archaea exhibit similar resistance. Some hypothesize that this resistance evolved as a defense against desiccation, extreme drying inflicting DNA damage similar to radiation exposure. Researchers have pinpointed specific enzymes within Deinococcus radiodurans, such as manganese complexes, that actively shield proteins from radiation's destructive touch. A 2019 study demonstrated the successful transfer of these radiation-resistant genes to other bacteria, opening a tantalizing possibility bioengineering life itself to withstand the harsh realities of space. From these microscopic marvels of natural resilience, a thrilling question erupts: could we engineer similar defenses into other organisms, crafting bespoke bacteria tailored for the extreme challenges of space? The answer resounds with a resounding yes. Genetic engineering unlocks a path forward. Scientists are now wielding revolutionary tools like CRISPR-Cas9 to precisely sculpt the genetic code of bacteria, effectively transplanting radiation-resistant genes from powerhouses like Deinococcus radiodurans into more versatile species. Imagine E. coli, the tireless workhorse of the biotech industry, now empowered with the ability to shrug off radiation's assault. A groundbreaking 2017 study at the University of Maryland vividly demonstrated precisely this, showcasing dramatically improved survival rates in modified E. coli following intense radiation exposure. But ambition soars even higher. Synthetic biology aims to design entirely novel proteins and metabolic pathways that surpass even nature's ingenuity. One promising avenue involves engineering bacteria to hyperproduce melanin, the pigment that shields our skin from the sun, but in vastly amplified quantities. This biogenic melanin could then serve as a potent biological shield, either within a self-contained bioreactor, or as a protective coating for spacecraft, greedily absorbing radiation before it can threaten astronauts or delicate equipment. The pioneering Biosentinel mission, carrying resilient yeast cells into the depths of space, is already beaming back invaluable data to fine-tune these strategies, paving the way for a future where genetically engineered bacteria form a living vanguard against the unforgiving radiation of space. Envision spacecraft exteriors shielded by a bioplastic infused with Deinococcus radiodurans, a bacterial marvel capable of withstanding an astounding 1. 5 million rads, 3,000 times the lethal dose for humans. Studies, such as the 2021 publication in PLOS One, demonstrate the feasibility of employing bacterial melanin to fabricate effective radiation shielding materials. Yet, the protective potential extends far beyond mere external barriers. Scientists are also exploring the potential of radiation-resistant bacteria as ingestible probiotics, tailored specifically for astronauts to fortify their internal defenses. While in its nascent stages, this innovative approach demands meticulous testing at specialized facilities such as NASA's Space Radiation Laboratory, NSIL, to guarantee safety, efficacy, and the prevention of unwanted mutations. Should this avenue prove successful, such probiotics could substantially mitigate radiation damage, paving the way for extended mission durations. Furthermore, drawing inspiration from the ISS's EXPOSE experiments and the astrobiology publication chronicling Bacillus subtilis's remarkable Martian survival, we begin to perceive that long-term bacterial protection on Mars is not merely a fanciful dream, but a tangible possibility. Yet, even as we envision bacteria as our cosmic guardians, shadows of doubt emerge. The Committee on Space Research, COSPAR, has established planetary protection guidelines, but are they enough? Introducing genetically altered organisms into the delicate balance of space could unleash unforeseen ecological consequences. A study in astrobiology even suggests the potential for gene transfer to hypothetical Martian microbes, rewriting the rules of an alien world before we've even begun to understand them. These possibilities deepen the ethical quandaries. Do we truly have the right to reshape celestial bodies to our will? The long-term stability of these modified organisms remains a significant question mark, leaving them vulnerable to unpredictable mutation. The future of space travel may hinge not solely on bigger rockets or faster engines, but on the very essence of life itself. Envision spacecraft walls alive with activity, a vibrant, living shield composed of Deinococcus radiodurans, not merely deflecting radiation, but actively mending damage, and even crafting essential resources. NASA's Gene Lab and missions like Biosentinel are setting the stage, unlocking the genetic secrets of radiation resistance. Melanin-rich fungi, transforming radiation into energy, might become the power source for our future voyages. Synthetic biology may empower us to engineer the ultimate spacefaring bacteria, ushering in a new epoch of sustainable and secure exploration, igniting hope for a boundless future among the stars. The ingenuity of science now turns toward these microscopic champions. Conan the Bacterium boasts an extraordinary resilience, offering us a remarkable blueprint. Imagine genetically engineered bacteria, fortified with melanin and radiation-resistant genes, standing as living shields. Contained within bioreactors, they could forge safe havens on spacecraft or even construct future Martian habitats. Inside NASA's labs, these biological bulwarks are undergoing tireless testing. The dream of Mars, once a distant horizon, now shimmers with possibility, carried on the backs of the smallest of allies. The promise of genetically engineered space bacteria as a biological shield is a radical departure from traditional radiation protection methods, offering a potentially sustainable and self-repairing solution for long-duration space missions. This innovative approach not only addresses the immediate threat of radiation exposure, but also opens up possibilities for resource utilization and habitat construction on Mars, fundamentally changing our approach to space exploration. Considering the potential risks and rewards of genetically engineering organisms for space exploration, what safeguards should be implemented to ensure responsible innovation and prevent unintended consequences on both Earth and other celestial bodies? Share your thoughts in the comments below.